Hello, everyone. My name is John Hughes, and I will be presenting on influenza virus A H1N1. An important thing to note before we get started, I wasn't aware of this, maybe you guys were, but I came into this thinking that H1N1 was only prevalent in the past pandemic, like the 2009 swine flu. However, I quickly found out that it is also one of the main culprits for the seasonal flu. So important thing to notice before we get started. Let's talk about the classification. It is of the family Myxoviridae, genus Orthomyxoviridae, and of the species Influenza, which can be subcategorized into 4 different subspecies, A, B, C, and D. When we're thinking about these seasonal outbreaks, we're mostly thinking about A and B, a little bit of C as well. C only contributes mild symptoms to only those ages like newborns to 2-year-olds. So it doesn't affect them that much. So it's not as prevalent as A and B, which cause those mild to severe seasonal flus, with influenza A being the cause of a couple of the past pandemics that we've experienced. As far as I know, influenza D is only responsible for some flu viruses that are expressed in cattle. And then H1N1, the strain we'll be looking at today, is under the subspecies influenza A. Let's talk more about the classification of influenza A and what exactly do the H and N mean in H1N1. They refer to surface proteins on the virus that assist with replication. You have your hemagglutinin, there's 11 known types, and then your neuraminidase, there's 18 known types. This little graphic right here just shows how scientists designate these strains. So, for example, in this example, you have virus type A, influenza A. It was first isolated in Sydney, Australia. It is strain number 5, isolated in 1997, and of the virus subtype H3N2. So, important thing to notice here is that there are different strains other than just H1N1, which can cause these flus. You have, as you can see here, there's the H3N2. You can also have H1N5. So that's an important distinction to make, that it's not just H1N1 that's causing these symptoms. And then up here, we have a pig and a bird. These are 2 of the main populations where these viruses can come from human or come into the human population. Some of the characteristics of H1N1, it is surrounded by an envelope, so it's going to have a lot of those hallmarks of a lipid membrane. You're going to have that lipid bilayer, some ion channels for nutrients to get into, and then you're going to have some surface proteins. Again, you have your neuraminidase and your hemagglutinin, as well as some other assistive proteins as well. And then inside the envelope, we have our helical capsid, which is surrounding our antisense single-stranded RNA virus. So what is the pathogenesis of this disease? So we have 2 transmission methods. We're going to have direct transmission and indirect transmission. When the virus is entering humans from animal populations, it's mostly going to be that direct transmission. But then when the virus is in a human population, it can be either a direct transmission or it can be an indirect transmission through droplet nuclei. So it's going to enter in through the nose and the mouth, and it's going to attack human alveolar epithelial cells and alveolar macrophages within the lungs and the major respiratory tract. The hemagglutinin is going to adhere itself to celiac acid molecules on the cell membrane, which is going to allow the envelope to fuse with the membrane of the host cell and enter into the host cell, where it can shed its membrane and then start its replication cycle. One of the more interesting things I found while doing this was that neuraminidase, one of the other major surface protein, actually assists the virus while budding. So the host cell has those celiac acid molecules on the host cell will actually adhere to the newly replicated viruses to try to stop them from infecting other cells. But the neuraminidase protein will actually cleave that connection to the cell and allow it to infect other cells. Let's talk about more of the pathogenesis. So the incubation period is going to last about 1 to 4 days, while you're going to be contagious 4 to 7 days. That first day is usually 1 day after it started incubating, and then after you've incubated it, you're going to start showing symptoms around 3 to 7 days since you first were in contact. And you're going to have those major symptoms around days 3 to 4. However, you can still see some lingering side effects. You could have a cough for like up to 2 weeks. So yeah, it really just depends person to person. But though about 2 to 3 days of serious symptoms are what most people experience. Let's talk more about the symptoms. So most of us with healthy immune systems are only going to experience those common and mild symptoms that I'm sure we're all very aware of, including fever, cough, body aches, vomiting, and diarrhea. However, those more at risk, and I'll talk more about the populations at risk in a few slides, they have a chance at contracting more severe symptoms, which include breathing complications, dehydration, which can be in the form of excessive diarrhea or excessive vomiting. That inability to breathe is going to contribute to more sluggish behavior. In some cases, you could experience a fever with a rash. And then one of the more severe symptoms, which is one of the main causes of death, is going to be that extreme leukocyte production, which will try to fight off those infected cells in the lungs. So they'll actually kill those cells, which will lead to suffocation and eventually death. So how does the body defend itself? So it does have its humoral response is going to be through the antibodies IgM, IgG, and IgA, and it's going to produce CD8 plus T cells to kill those viruses as well through apoptosis and cell lysis. The vaccines that are currently being used right now are obviously going to incorporate these antibodies. However, researchers have been trying to push towards utilizing these CD8 T cells more, just because of the surface proteins of the viruses, the hemagglutinin and the neuraminidase, are very likely to mutate very quickly. I mean, we can see with like COVID, there's been like 3 different mutations very quickly. So those RNA viruses can quickly mutate, possibly rendering those vaccines useless. So those CD8 plus T cells, they're looking into how can they protect better against mutations and antigenic drift that the virus will undergo. All right, back to the populations at risk. So with traditional flu endemics, it's going to affect mostly adults 65 years and older because of their weakened immune systems, and then children ages about 0 to 5 because of their undeveloped immune systems. However, one of the more interesting things that was noticed during the H1N1 outbreak is that it mostly affected those 65 years and under. The 2009 H1N1 virus was this conglomerate of different strains from avian, swine, and human populations, which older people had seen more in their lifetime, so they had developed an immunity to some of these strains. However, those 65 years and younger weren't in contact with those strains, so there was more of a chance that they would have severe symptoms. Those struggling with obesity are also going to be more at risk for severe symptoms, and then the immunocompromised, those with underlying health conditions, pregnant women, people like that. So how do medical professionals diagnose the flu? It's pretty easy. You just look for the common flu symptoms, especially in flu season. If they see you with a cough or a fever during the flu season, they might just send you home and just say, drink some more water, get some more rest. However, if they're unsure, there are tests that they can administer, by introducing saliva into one of these machines. They can just tell real quickly. It takes about 20 minutes. They can figure out which virus you have, if you have any. These are rapid flu tests. Then treatment for the flu, it's usually pretty simple. You just eat more, drink more, sleep more. You should be good in a few days. However, with severe symptoms, it might be necessary to take some antiviral drugs. I'm sure most of you are aware of Tamiflu and then Relenza as well. These are 2 drugs that can assist with the symptoms. So how can we prevent H1N1? So the main way is through vaccination. A large percent, as you can see here, of the population are vaccinated yearly. There's two different types of vaccinations that people can be administered with. There's the attenuated version and the inactivated version. Attenuated just means like a weakened version of the virus that can't really cause any symptoms or anything, but still gets that immune response to act against the virus. And then inactivated viruses, these are just dead viruses that will still start an immune response. And then also just the classic things that we should always be doing. Make sure you're washing your hands well, covering your coughs when you need to, avoid touching your face, making sure to clean surfaces. Viruses can last up to about 24 hours on inanimate objects, so make sure you're cleaning those surfaces, and avoid large crowd contact. These are all ways that are encouraged and promoted by the governments of the world to stop the spread of flu. Another important factor is having access to prevention and treatment resources. I'm going to show some statistics later, but during the 2009 swine flu, a lot of countries in East Asia and East Africa were heavily affected because they weren't able to get vaccinated, or they didn't have access to clean drinking water, clean resources, so they had more of an effect for getting that virus. Let's talk about the historic perspective of H1N1. So influenza viruses have been around basically since man has been around. There have been manuscripts from Hippocrates who reported symptoms, flu-like symptoms. However, the main H1N1 didn't present itself until 1918, where it presented itself in a big way with the Spanish flu. This was from an avian genome. I'll talk more about the statistics of Spanish flu, but that was the first time where it was introduced into the human population. Then in the 1930s, you have the close study of H1N1. People were able to isolate it, determine it was a virus, determine it was the cause of the Spanish flu. Back then, people thought it was a bacteria. They weren't really sure what it was. People are able to create vaccines for it, and just learn more about the characteristics of H1N1. The next major event is the Fort Dix outbreak in New Jersey. In an army base, they had about 13 people get infected with H1N1 with severe symptoms, and one person die, which began a national effort to get people vaccinated. They were able to get 25% of the population vaccinated. I'll talk more about that in just a second. And then the next big event is the Russian flu. This is more of a conspiracy theory than anything, but the flu virus that was circulating that year was noticed to have similarities to a flu virus from the 40s and the 50s. And this was during the Cold War, so people are thinking that Russia is trying some form of biological warfare. And then the next major event is the 2009 swine flu. Let's talk more about the 1918 Spanish influenza. So this was had more deaths than World War II, with 50 million deaths. It had 500 million cases, which is a third of the world population. And in the U.S., it had 675,000 total deaths. I thought this picture was kind of cool. How even back then they were showing people to wear a mask, so that's kind of fun. And then in 1976, again, we have the Fort Dix outbreak, 13 cases and 1 dead. This also led to some conspiracy from Americans who thought that it was faked by the government to institute a national vaccination effort. And it was caused by a novel H1N1, which is mutated and still, if we can see here, it's actually still in circulation today. It's a mutated form is. And then the 2009 H1N1 pandemic. This began on April 12th, 2009, and it ended in April 10th, 2010. In the United States, it was mostly widespread in California, Maine, and Hawaii, with 60.8 million total cases, 305 hospitalizations, and 12,469 deaths. And then worldwide, it had 151,700 to 575,000 deaths. That number is so drastically estimated is because they weren't really sure how many people in Southeast Asia and Africa were actually dying. Again, these prevention and treatment resources were very limited. And then this disease first originated in Mexico and the first strains were found in California. This picture right here is actually from my elementary school. We had a few of the first outbreaks in probably in North America at the time, and we got a couple weeks off from school. So third grade John was very happy about that. This again is just a map of the swine flu and it's how it affected the world. So we can see here in Asia, in parts of Africa too, we're going to have more prevalence and more deaths than more populated and more developed countries. And you can see in South America, there were also a high number of cases as well. So what about H1N1 now? So interestingly enough, the cases are at an all-time low because of efforts against COVID. So in defending against COVID, people are also defending against H1N1 in the annual flu virus. However, new strains are constantly evolving. This strain right here is one that both of these strains are in circulation right now and are the cause of seasonal flus that we're seeing. And then researchers are also working on mechanisms such as these algorithms, which explore the probability of outbreaks among avian populations. So, like, how likely is it that a new novel H1N1 is going to break out, as well as where would it go if it was to break out. These are my references, and thank you very much for watching.